to ace the test. Okay. So let's focus here on what you need to know here. Let me get my voice back up on here, because sometimes the sound fails. So trust the sound, it's working today. So looking at the enzymes, we're going to know what you need to look at catalysis, and then you're going to know what is happening with systems, with pathways, and how it is that cells can manage for enzymatic pathways and enzymatic reactions go a certain way. So what happens here? These enzymes are mainly proteins. Not all of enzymes are proteins, but most enzymes are proteins, and they are going to bind their substrates, and they're going to make products out of it. The substrates are always going to bind at the active site. That should not be any news for you, right? So then what happens here? You have the enzyme that gets together with the substrate. Then you're going to make an enzyme and substrate complex. That complex is going to do its magic. Then you're going to end up with an enzyme and products complex. And from there, you're going to spill out the products, and you're going to recover the enzyme at the original condition. So one main thing to know here, enzymes do not get consumed in the reaction. They are going to be spilled out the same way that they went in. If there is any modification to the enzyme might be useless afterwards. Okay? We're going to look at a bit of modification later on after you digest all the enzyme here. All right, so what happens here? The enzyme is going to reduce the amount of energy that you have to put in order for the reaction to occur. What is it the enzymes do? They're going to put the substrate in the right conformation to increase the probability of the reaction that is spontaneous, increase the probability of that spontaneous reaction to happen. Enzymes are not going to do a miracle. If the reaction has a positive delta G, the enzymes are not going to make the reaction happen. Okay, the reaction is not going to happen. So it cannot. The enzymes are not going to do things that are against thermodynamics law. Okay, these enzymes are going to stabilize the intermediates so that they push this thing to go towards product. Okay, so mainly what the enzymes do is speeding up this whole process of the naturally occurring reaction to go on. So what kind of reactions you can have? You can have dissociation reactions in which your substrate gets broken down. You can have association reactions in which you put things together. Okay. And when you look at what enzymes are doing, in case here of dissociation, you have a certain level of dissociation rate. So without the enzyme, this thing is going to take a long time to dissociate. But then with the enzyme, you have a bit of a faster dissociation rate. Same thing with the association. Now, let's say that if you have reactions in equilibrium condition, okay, so the rate of getting together with the substrate and separating from the substrate, meaning the rate of going in and coming out, on and off, they would be the same. So at equilibrium, you have association is equal to the dissociation of whatever that enzyme is doing. Okay? So that rate of going on and coming off, that rate at equilibrium is called the equilibrium constant. Okay? And that is associated with the ability of the enzyme to bind to substrate in case of enzyme kinetics. And here you notice that I added this little video from Jove, right? So take a look at that. This video is not deriving the Michaelis-Menten equation, but it's giving you a nice visual idea of what is happening here, which is actually quite simple. All right. So enzymes are usually very, very fast. They're very, very good at what they do. And based on evolutionary principles, enzymes get to be so efficient that any modification in the structure, in the primary amino acid structure of these proteins, any modification would actually make them a bit slower, okay, because they are really at the fastest rate and most useful rate that they can have. All right. So they hold the reactants in proper orientation, and what that does is that the electron clouds that are involved in covalent bonds, they actually get to be very close to places where they can be distorted or changed or temporarily exchanged with amino acid or a residue in the enzyme. So we're going to look here at examples that you absolutely have to know mechanistic pathways. Okay. So they're going to bind and they're going to stabilize the intermediate. A lot of times, the intermediate is highly reactive. And some other times, the intermediates are actually toxic to the cells. So this enzyme pathway has to be sequestering these intermediates. And they have to isolate the intermediates from the rest of the cytoplasm. Because if these intermediates will fall out of the enzyme, they can actually kill the cell. Okay, and this we actually see in pathways like urea formation and manipulation of ammonia. Okay, so you can imagine that this is true not only for eukaryotic cells, but it's also true for prokaryotic cells, right? Because prokaryotic cells undergo a lot of the same metabolism that we see in eukaryotic cells. Right? So if these intermediates are just losing the cytoplasm, then it's going to kill the cell. Right, so what these enzymes do, they're forming this little cage or this little tunnel that keeps the intermediates stuck in one place, so they cannot really harm the cell. Okay. So here, important things to know. Okay, we mentioned this. Enzymes are not going to change the delta G. Okay, there are several types of reactions that you can have. You can have one substrate that is changed into something else, but it's the same substrate. So this would be the case of an isomerase type of reaction. Okay. You can break down, or you can build up substrates. You can break down substrates. So you can have synthesis or breaking down of the reactants, or you can exchange groups in substrates. Okay. Now keep in mind that enzymes, they always work forward and backwards. They will stabilize the intermediate. So the Chatelier principle also applies here, meaning that if you have a lot of products and you're stabilizing the intermediate, most likely you're not going to get product out of that intermediate. You're most likely going to shift back to the reactants. Okay? Cells usually sequester the products of reactions. So in that way, this type of reaction will always be forward, be pushed to the right. Okay, so here are examples of enzymes that are not proteins. So we say ribozymes are examples of.
Does that bond on say H3?
Yeah. You can see over there? Is it this thing blocking?
Oh, yeah, I can see it now.
And you also have in the PDF that I uploaded. Yeah?
Steve said that they go both directions.
Yeah.
But then
Usually the products get pushed out after the products are made. Yeah, they leave, meaning that if you have reactions in series, the next enzyme will catch the product from the first enzyme. So you're always having deficit of product, so the reaction is pushed to go in one direction. Okay, so what is happening here? First thing that happens, the enzyme is happily sitting there, and let's assume that these little arms are the active site. And here is just a picture to understand what's going on with the enzyme. Usually the active site is like inside of the enzyme, it's not like exposed to the outside. So first thing that is going to happen is going to bind the substrate. Binding of the substrate is going to change the conformation of the enzyme. So the enzyme goes from that to a different conformation, and that kind of conformation is what's going to allow these substrates to interact or the reaction to happen. Once the reaction happens, these things that are bound, let's say the products, they are going to have different electron clouds. So the products themselves are going to push the enzyme back to the original conformation. Then the products are going to dissociate. And then you end up with the enzyme exactly in the same shape that you had before. Okay, so here should come your first question. Let me see if I can finally get this thing to work. I'm going to try to go through the website and see. So let me pause this recording. What? Yeah, it's, for some reason this thing is not working, it's not connecting. I don't know why. Anyway, I'll put these questions in the next class and you can recover it then, okay? So let's just keep on going here. So here it says, during an enzymatic reaction, the substrate binds to each of the following. What would be your answer here? The active site, yeah. So keep in mind, next time you get double points. All right. So here, coming back to your enzymatic reaction, what is going on? What is it you need to know? Here, you're going to have what the enzyme is doing. It is lowering the activation energy. And remember, you had one exam question asking you what was the delta G of the reaction. Right? So now we're getting fancier. We're going to see what is happening with the enzymes that are actually lowering the activation energy, but they have nothing to do with the change in the delta G. Okay, so here, looking at considerations for kinetics of what is happening with the enzyme. Remember, enzyme is a protein, and you have to take in consideration how much protein you have in relation to how much substrate you have. If you have just a little bit of substrate, then you're not going to have a lot of chemical reaction going on because you don't have much to work with in the beginning, to start with, okay? And also, keep in mind that not only the amount of substrate has to come into consideration, also the concentration of the enzyme, you have to keep that in mind, okay? So let's say here that you keep the concentration of your enzyme constant. You don't want to change two variables in one time, right? So you keep the enzyme constant, and what you're going to do here, you're going to increase the amount of substrate that you have available. So as you increase the amount of substrate, you are going to get more of your reaction happening up to a certain point. Because if you put just too much substrate, you're going to saturate your enzyme and you're not going to get any more of your products being produced at one time. Because you can compare this with a bakery, for example. You want to give the substrates, like flour and water and yeast, and you have only two bakers working on your bread, right? So you give a little bit of flour and then water and yeast, and your baker is like, one of them is making the bread, the other one is relaxing on YouTube, watching TV or whatever. You give enough substrate and both of them are working. You give it too much and they cannot produce more than what they can manage, each person. So saturation based on amount of substrate. Yeah. Sorry? Oh, how is the amount of substrate increasing? You can do this artificially if you're running an experiment, or you can have a certain pathway that have the raw materials available in order for the enzyme, for the reaction to happen, okay? So in this case, you can say artificial experiment. You have 20 tubes in series. Each tube has the exact same concentration of enzymes. And then in the first tube, you put a little bit of substrate. Second one, you add a little bit more. Third one, you add a little bit more. And then you put a dye to recognize when your reaction is going to happen, okay? So you're just counting what is the speed of your reaction and how much it will take for you to get saturation of your enzymes. So this is what is happening in a real enzymatic reaction. At first, you see that you have substrate that is not converted into product, so you have a lot of substrate. At the same time, you don't have product in the beginning, so no product, close to zero here. What is going to happen? You have a lot of enzyme that didn't bind yet. So what's going to happen, as soon as you add everything together, you're going to start having enzyme that binds. So enzyme and substrate is bound, it's binding. So your free enzyme is going to go down. You're going to start getting your product that is being produced. So your product line is going to go up. At the same time, you're consuming your substrate, so your substrate is going to go down. So this area over here of a lot of change, this is kind of difficult to work with mathematically. Okay? So we don't deal with that area. We go to the simple stuff. Okay? All right. So as the reaction is going on, you're going to have one area here in which you're going to have very little change in free enzyme and enzyme bound. Okay? And then later, you're going to have an area in which you're going to get your free enzyme, you're going to get a lot of the products, and your substrates are going to disappear because everything has been converted into product. So this is all this graph is telling you. So what do you want to deal with? We don't want to deal with pre-steady state conditions because that's messy. You don't want to deal with the end point because that's also messy. So you want to deal here with this stretch that is considered a steady state condition, and that is going to take in consideration that your amount of enzyme bound is not changing much. The amount of enzyme unbound is also not changing much, but we usually take in consideration more of the enzyme that is bound because that's the one that is doing the work. Okay? So this stretch here is going to be your steady state condition. Now, what is being plotted here? That is concentration of the stuff over time. Okay? So this time here, when concentration of bound enzyme doesn't change much, that's what you call your steady state condition. Okay. Now, how are you going to see how much substrate is consumed? Well, you're going to look at your substrate line. Yeah, sorry, there are inverted colors here, but substrate line, here's your substrate. You start with high substrate, you end up with low substrate. If you plot here this line, substrate over time, and you take the derivative of that line, remember derivatives? Who took calculus? Everybody should have taken calculus? Who never seen derivatives in your life? Who doesn't want to say? Okay, I think calculus is fun, but I'm not going to torture you too much. So what's happening here? How are you going to figure out how quickly this thing is going? You're going to take the derivative of that line, okay? So you're going to say change in the substrate, that's your y-axis, over change in time, that's going to give you the velocity of that line. It's also called first order kinetics, okay? All right, so in comparison to products are increasing, and we're going to work with that little stretch in which the product is increasing linearly. So your equation is actually simple. How fast is the product getting made? Well, you're going to take the derivative. You're going to take derivative of the product, that is the y, change in y, over change in x, dp over dt, and that is a positive line, right? Is you're producing products, that is going to give you the velocity of formation of your products, okay? This should be actually quite simple. All right, so what are we going to deal with? We are going to deal only with that little area in which this line looks straight, okay? So when this thing is going relatively at a straight line, like it is over here, that's what you call first order kinetics, okay? Derivative of the line gives you the speed of the reaction, that's all. Now, we know that the whole thing, as you increase substrate, is not going to keep on going up, because it depends on proteins. and proteins are a certain number in the cells, and once you saturate them, they cannot do any more work. Okay, it's like overloading those, bakers, right? You produce more bread, and there are only 2 people. Okay, so here, as you saturate your enzyme, you're going to have this plateau, and you're going to reach a maximum velocity. That doesn't matter how much more substrate you add, your velocity is not going to increase because all your enzymes are already working. Okay? So you have to wait until the enzyme is done for the enzyme to pick up another substrate. So you're going to have this maximum velocity, maximum production of your product. Okay? When this plateau happens, this is when you call a zero-order kinetics. But you're not going to be really worrying too much about that. Okay? We're just going to know that at this point, all of your enzymes are already working at full capacity. Okay, so your speed is not going to increase by anything. Okay, so we're going to be worried only about this side here, or of a straight line. So equation gets to be simple. Okay, everybody's cool with that? Yeah, yeah. Have you seen enzymes before? Took some biochemistry? No? It's really, really easy. Okay. So what is happening here? This is the part that gets to be a little tricky. So this is the part when I really need your brain power to be at the maximum, okay? So let's increase your brain power. Put some oxygen going to your brain. How do you do that? You can breathe, you can yawn, that's going to put you to sleep. Lift your arms, up, go. 3 times. Enzymatic reaction, get oxygen in your brain. Good. Now, focus on this. This gets to be tricky. You gotta know this. It's okay. All right. When your enzyme is saturated, you get your maximum velocity. That's your Vmax, right? Right. When your velocity is half of your Vmax, that half is going to correspond to a certain dot on this curve, okay? It is for sort of kinetics, so it's a little curve because an enzyme, you know how that goes. All right, so what is going to happen here? That dot on the y-axis here, you're going to drop it and find the concentration of your substrate on your x-axis. That concentration of your substrate is going to tell you how good the enzyme is at binding your substrate. Okay? So here, that concentration of your substrate is going to give you this value that is called Km. Km is going to give you a value that corresponds to half of your maximum velocity. Okay? Now, let's see what this means. If you have an enzyme that is not good at binding substrate, what do you need to do for this enzyme to bind the substrate? You need to give it a lot more incentive. You need to give it more substrate for the enzyme to bind. Okay? So if the enzyme is not good at binding substrate, so let's draw here a line, and I wish this pen was working. Yeah, it's working. Okay? So let's draw here a line that is going to tell you the enzyme is not good at binding substrate, but it's going to reach the maximum velocity that is the same as this other enzyme that is better at binding substrate. Okay? So for you to get to the maximum velocity, let's say that, okay, you need to get this much substrate concentration. So let's say, let's give it a number like 100 millimoles or something. Okay? Now this red enzyme, it reached the same Vmax, but look at this. The half of the velocity, the half of Vmax for this slow enzyme here is going to be right there. So the half of the Vmax, if you drop this line, is going to be right here. So let's say this is like 50. In comparison to your good enzyme that picked up substrate very quickly, your good enzyme reached a Vmax at about 25. So you needed less substrate for your good green enzyme to work. So we say that a low Km makes it a better enzyme. Okay, so low Km means your enzyme can grab to substrate quite easily. If you need a high Km, that means your enzyme doesn't really like to bind substrate very readily. Okay? So this is a part that is really important to understand here. The Km is telling you the concentration of substrate that your enzyme needs in order for it to work. Okay? Now, this is a part that gets tricky because here it says, so Km is not the same as Vmax over 2. Km is not the same as half of the velocity. Okay? So the question here would be, why? Why Km is not the same as half of the velocity? Any brave soul? Yeah. It went down. Yeah, because depending on your enzyme, you can have different Kms, but if you have the same maximum velocity, then they should all be the same. So maximum velocity and half of the velocity is not really telling you exactly about the Km. When you do the intersection at your substrate versus velocity line, this is when you're going to find out what your Km is. Okay, so let's put here another enzyme here that is even better than the first enzyme, and it can bind substrate very, very quickly. So let's say your enzyme would have something this. So the Vmax for your blue line here would be the same Vmax as the other two, but now if you go half of Vmax, it's going to be here. So now your Km is going to be like right there. So let's say it's around 10 instead of 25. Okay? So this blue one, is it going to have higher affinity for substrate in comparison to your green line, or it's going to have lower affinity? It's going to have higher affinity because you only need a little bit of substrate for it to reach your half maximum. So is that clear? X times your substrate over Km plus your substrate. Okay? So this is plain Michaelis-Menten type of equation. Okay. All right, so the question here was saying in reaction rate versus substrate. The substrate concentration, a half of Vmax, is represented by what? So here you should say Km. Right, make sure you understand that. Okay. So types of enzymes. It's really easy usually to identify what the enzyme is doing, because the enzyme is going to tell you what it's doing. If it has A's at the end, so A's is a telltale sign that this thing is an enzyme. Okay, so if you say hydrolase is using water to do something, to break something. So this is in general a hydrolytic enzyme, okay, that is going to cut something. Nucleases is breaking down nucleotides. Proteases are breaking down proteins. Synthases, what are they doing? Synthesizing stuff. Isomerases are shifting things in one molecule, shifting things around. Polymerases are usually concerning nucleic acids, so building up nucleic acid chains. Okay? Kinases and phosphatases, those guys you really have to know. Now, oxidoreductase, oxidation reduction, ATPases, they're going to make ATP, GTPases, they're going to break down GTP. So you look at the name of the enzyme, you should understand what the enzyme is doing. All right, so they have classifications for those enzymes. Don't worry about these EC numbers because I can never remember them. But my rule is that if I cannot remember the stuff, I'm not going to ask you about it. Okay? But you have to know the names, what the names are indicating. Oxidoreductase, oxidation reduction. Transferases, they're going to transfer a group, and so on and so forth. Okay? All right. So here, example of enzymatic reaction that is very classic is lysozyme. So this is the guy that I have changed a little bit in your PowerPoint to make it look a little easier. Okay, so lysozyme is a type of enzyme that is going to break bacterial cell wall. So it's going to protect you against invading bacteria. So this lysozyme is going to use water, it's going to do hydrolysis, and it's going to break that sugar chain in the bacterial cell wall. Okay. Overall, the energy of the delta G of the reaction is favorable, but it just takes a long time. Now, the problem is that in order for it to happen naturally, you have to put in a lot of energy, so high activation energy. What is the enzyme going to do? It's going to shift that bond, and it's going to make breaking that bond easier. Okay, so here's what's happening. This is a bacterial cell wall. It goes in the pocket of your enzyme, of your lysozyme. Okay. So the lysozyme binds to this polysaccharide chain, okay? And it's gonna do something. First thing it's gonna do is that it's going to distort this sugar in a certain way to make it look uncomfortable, okay? So let's see what's gonna happen here. The thing gets distorted, and you're going to have a nucleophilic attack. You're going to have actually here a nucleophilic attack of this oxygen that is going to steal that hydrogen from your glutamate. Glutamate is at the active site of your enzyme. Okay, so this oxygen, it has the electron cloud, is going to steal that hydrogen. So once this thing happens, this bond is going to break. Okay, so this bond is going to break, it's going to go that way. This carbon here is going to be partially positive. And it is accommodated in a certain way that is close to this aspartate that has a negative charge, also in the active site, but in the other side. Okay, so what's going to happen? This guy steals the hydrogen, this becomes partially positive. Now, this guy here attacks, this is a nucleophilic attack in this partially positive carbon right there. Okay? So now, you're going to have a covalent bond that is going to form between your aspartate and this first sugar molecule right there. So this here breaks that bond, this here makes another bond. Okay? So let's see what's happening here on the other side. So this is what happened. This guy stole the hydrogen. Oh, this is next step, okay? Okay, so here. This guy here first took the hydrogen from glutamate. So this bond here is a new bond. This sugar, number 2 here, is ready to go out. Okay? So this guy is done. The other guy here is going to get covalently bound. Okay, so where is it here? It's not showing the bond. Okay, there it is. This guy here is covalently bound to the carbon. Remember, carbon makes 4 bonds. It's tetrahedral, right? So aspartate here is bound to your substrate, to sugar number 1, and you count the number of bonds, you're going to see 1, 2, 3, 4 bonds. But if this guy stays there, that's a problem for your enzyme because your enzyme will be modified. Got covalently bound to something. You don't want this bond over there. So what's going to happen here, this is the part where you're going to have your hydrolysis. So a water molecule is going to go there. This guy here, they got the hydrogen stolen. It has this big electron cloud that can steal a hydrogen from the water. And it can recover its hydrogen. This hydroxyl group over here is going to do nucleophilic attack on this carbon and is going to displace that covalent bond. It's going to send it back to the aspartate. Okay? So what's going to happen here, you're going to put hydroxyl group in there, you're going to break the bond, and you're going to end up with your original aspartate that had a negative charge, your original glutamate that had a hydrogen, and you're going to end up breaking this bond that was in between this polysaccharide. Okay, so this guy here came from the glutamate, and those this hydroxyl over there came from your water molecule. So this hydrogen here also came from the water molecule. So at this point, your bond in your polysaccharide is broken, and you liberate your products. Okay, was that clear? Yeah, everybody's cool with what's happening here? Okay, so here is a little movie showing you how this reaction is happening. Right, so look, this is a part that I couldn't put in the figure, showing you that, see, the enzyme twists this thing around. See, here is conformationally stable. You see, look, now look at the R's here. They're going to be in conflict with this electron cloud. So this is the original shift that the enzyme does in order to put this carbon here and allow this other carbon to be targeted for the enzymatic reaction. Okay, look. This one steals the hydrogen. This covalent bond is temporarily made. Then you get your water molecule that replaces that hydrogen and breaks that temporary bond at your aspartate. Enzyme is recovered and your products come out. When the products come out, then this thing goes back to the original conformation. How is it that the thing flips? Because the ester bond is a single bond, so you can have rotation in a single bond. Okay. All right, so more in protein functions. We're going to look at allosteria. Enzymes use help from other molecules, and those can be non-protein parts of the enzyme. Sometimes you do have a little protein that can go and serve also as a cofactor. So you can have metal ions, or you can have some other type of molecule that goes in and helps the enzyme to work. Sometimes they are permanently bound, so you can have a covalent bond, but most of the time they are bound by non-covalent interactions, so those are your electrostatic forces. Okay. Usually they do not bind at the catalytic site. Sometimes they do, but most of the time they bind at some other site, and that's called your allosteric site. Okay. If your thing is inorganic, like a metal, then you call that a cofactor. If that thing is organic, you call that a coenzyme. Okay, so coenzymes are a type of cofactors. Okay, but they are organic. So most vitamins are actually coenzymes. If you do not have the vitamin, then your enzyme does not work properly. Okay, so types of vitamins that get transformed or are purely the cofactor for some enzymes. So we have them here. So we have here, for example, vitamin B1 and B2. Those are important for some types, we're going to see later, some types of reactions. All the B vitamins, right, and then you see here like B12, it gives rise to cobalamin enzymes. Those are very important for the proper functioning of those metabolic enzymes. And this is FYI, we're not going to ask you which enzyme is working on what in this cofactor. This is FYI, so just, don't panic. All right. So as I mentioned, you have formation of molecular tunnels that are going to keep those reactive intermediates in a certain orientation so that this process is going to happen inside this major complex and is not going to allow this toxic thing to fly off to the cytoplasm. Okay? It's going to protect your cytoplasm from diffusion of those toxic intermediates. The example here is of carbamoyl phosphate synthase. It's this huge complex of several enzymes that are working together to get your end product. How do you regulate the function of enzymes? All right, so enzymes have to be regulated. Sometimes you need some products, some other times you have plenty of the products, you don't want to have too much of those products, okay? So you can regulate how much of the enzyme you make. You can regulate how much of the product you're making. You can regulate how much of the protein you are breaking down. And you can also compartmentalize the protein and sequester it in a certain place in order to keep it inactive, but ready to get activated in case the cell will need it. So you have several strategies of control of reactions. So here we're going to look at direct control of the protein activity. Okay. So what is going to happen here? Okay, so main things that you can control. You can control whether the protein is going to bind your substrate. You can control the shape of your protein by phosphorylation. You can control whether the protein is active or not, and those are subtypes of enzymes. They're called GTPases or GTP switching proteins. And you can have physical control of what your enzyme is doing. Okay, so we're going to go through each one of those and give you examples of what is happening here. Okay, so you have your main active site of your proteins, of your enzymes. So this is where your substrates go. Now, the site that is regulating the protein, you call that your allosteric site. Okay. So let's see here what is happening here. When you put this other molecule that controls the function of your enzyme, you are going to call this allosteric regulation. So the presence or absence of that substance is going to put the enzyme into this allosteric transition. Okay, so let's see what's important to know here. Okay, what kind of molecules can be allosteric regulators? You can have other little proteins, or you can have ions, you can have organic or inorganic types of regulation. The thing can come from inside of the cell, or it can come from outside of the cell, like calcium, for example. It can go inside of the cell and allow some enzymes to work, or it can stop the function of some enzymes. So it can be basically anything here. All right. So let's see what makes sense for this type of regulation to happen. When you have a certain pathway of enzymes that are following each other, so this enzyme is getting substrate and is making a product that is being used up by this enzyme, that is making a product that is using up by this enzyme, and so forth, right? Now, in this type of pathway, the product of this enzyme can be used in two ways. It can go either that way towards C enzyme, or it can go down towards X enzyme. This is commonly found in synthesis of amino acids that are non-essential. Okay, so what's going to happen here? If you need this certain amino acid, then you are going to shift this product towards this pathway. But once your cell has enough of this product, you don't want to keep on making Z because it's going to be energetically wasteful, and cells do not like to waste energy. So what's going to happen is that this product is going to have a certain allosteric site at this enzyme here. So the enzyme is in the arrow. So let's put the enzyme over there. So this is your enzyme here. Okay? So this is your substrate for that enzyme. And now Z over here is going to have a little pocket that it can go back in here and bind to the little pocket. Once the Z binds to this allosteric site, it's going to change the conformation of this enzyme. It distorted the enzyme. And now this enzyme cannot bind the substrate anymore. Okay? So this is called negative inhibition, negative feedback inhibition. So the product inhibited the pathway for its formation. Okay? Now, a good thing for this product to do is to inhibit the first enzyme of its own pathway, because you don't want to be wasting energy making other intermediates if those intermediates are not going to be used for anything else. Okay? So this thing here, if it stops the first enzyme, is going to save energy, and you're not going to end up with these things in the middle that are not useful. Okay? So this example of feedback inhibition. Everybody got that clear? Cool? All right. So here is an example of what happens in case of amino acid production. So those are amino acids that are not essential. Most amino acids you make in your body by converting other amino acids. Some amino acids are essential, so that means you have to have it in your diet, right? So what is happening here? Look, you have this starting amino acid that is aspartate. Okay, aspartate is actually quite important amino acid. So look here, aspartate can be converted to something else, and then to something else that is an intermediate in two other amino acid pathways. So this guy here can go to lysine, and this guy here can go to homoserine, they can go to threonine, or can end up in methionine, and this thing can be modified some more in isoleucine. Look how many amino acids you have here. You have the original aspartate, then you have homocysteine, that is not quite very useful, but you have homocysteine. You have threonine, isoleucine, methionine, lysine, and you're thinking, how can I control all this mass? Well, it depends on what your body is using. If you need to produce more lysine, for example, okay, let's say your body is fighting a viral infection, you want to produce lysine. So what's going to happen? You get your aspartate, you're going to go this pathway, you're going to go down this way, and you're going to go in a pathway to produce lysine. But then look what happens. This one here, you're not only using this intermediate to produce lysine, you can also use this intermediate to produce something else. So if you want to produce lysine and you have enough concentration of lysine, you want to inhibit the production of lysine at the first enzyme that is useful only for the production of lysine. Okay, so lysine is going to do feedback inhibition in the first branch of this pathway that is useful for lysine production. At the same time, lysine is also going to start to inhibit the production of the first intermediate. Look at this. It's working here and it's working over there. But now you say, wait a minute. If I have this enzyme here that is important for the other amino acids, do I want to inhibit completely? The answer would be no. Because if you inhibit completely, then you're going to stop production of the other amino acids. So inhibition of lysine is only partially going to inhibit this first enzyme. Okay? But it's going to have enough of the enzyme work that is going to be sufficient to produce the other amino acids. And that is possible because the Km of these enzymes that come afterwards, they have more affinity to their substrates. Okay? I know that gets a bit of a too much wordy kind of thing. Now, look at this. 3 of those amino acids are working directly in the first enzyme to block this flow. And the others are working in specific pathways that are going to block the first enzyme of their specific path. Okay, so this is an example of negative feedback. Everybody got cool negative feedback? Yeah? No. Am I going to ask you about negative feedback? Yes. On the other hand, you have some enzymes that work with positive regulation. So it's a positive push of regulation, not a feedback, but it's a positive regulation. Yeah, here it says positive feedback. Okay. So what happens here is that sometimes you have accumulation of certain product, for example, here is ADP, that will bind to an enzyme and is going to push it to work. Okay. So here is saying as the concentration of your ADP increases, that is going to push oxidation of sugars in your metabolic pathway because you're going to need to produce more ATP. So increasing level of ATP is going to push production of ATP. So it's actually going to activate some enzymes. So this type of regulation can be, the other one is feedback inhibition. So this one is positive feedback, depending on the presence of this allosteric factor. Okay. All right, phosphorylation. Phosphorylation is going to bind covalently phosphorus to your enzyme. There are only 3 amino acids that are going to receive phosphorylation. Put them here, there they are. Okay, so here is your enzyme, and you're going to have one of these 3 amino acids, and they can be either serine, or threonine, or tyrosine. Why? Well, because they have groups that can accept your phosphate. They have hydroxide groups that can accept your phosphate. Okay? So what's going to happen here? You're going to use ATP as your source of phosphate, and you're going to use an enzyme that you're going to put a phosphate on your protein, on your enzyme. Okay? The name of the enzyme that puts phosphate is kinase. So kinase will add a phosphate. The addition of the phosphate is going to change the bulkiness of your enzyme, and it can either activate it or deactivate it depending on the enzyme. Some enzymes get activated by the presence of phosphate, some other enzymes get deactivated with the presence of a phosphate. So you really have to look at the pathway and what the cell needs. Okay? So kinases will add a phosphate, and the phosphate is always going to come from ATP. Okay? ATP is needed for that. If you want to take the phosphate out, then you're going to use this other protein that is called the phosphatase. Phosphatase will break away that phosphate, and it's going to give your enzyme without the phosphate. Sometimes it's going to activate, sometimes it's going to inhibit. It really depends on the enzyme you're working with. Okay, so what are the 3 residues that can accept phosphate? Serine, threonine, or tyrosine. So sometimes adding a phosphate will turn the enzyme on. Sometimes adding a phosphate will turn your enzyme off. And here would be another question, but of course it is not working. So then we get to this last part here that is talking about GTP controlled enzymes. Okay? So what is happening here? When the enzyme hydrolyzes GTP, you call that a GTPase. Okay. And that is really, really important for signal transduction, for whatever the cell is getting about information on the outside. You're going to see later these types of signal transductions. Okay. So what is happening here is that the presence of GTP that binds to this enzyme, the presence of GTP here is going to activate the enzyme. And it's always like that. If you put GTP, the enzyme is going to get activated. If GTP gets hydrolyzed to GDP, the enzyme is going to be quiet. Okay? And the presence of GTP bound is is temporary. It's not going to be there forever. Okay? So what is happening here? When you have GTP bound, your enzyme is active. When you have GDP, your enzyme is inactive. Okay. How is it that you're going to get GDP out of this? Well, the enzyme itself sometimes does the work. So the enzyme is also a GTPase, and that means it's going to break a phosphate out of the GTP. So the enzyme can do that over time. So we call that a GTPase. Right? So what is happening here? So let's see here. You have your enzyme bound to GTP and it's active. This enzyme can hydrolyze GTP and end up with GDP, and then your enzyme is inactive. Okay. Now, GTP stays stuck in the enzyme for some time, not forever, because it's not crazy glue. So it's association and dissociation. Sometimes it will dissociate. When GDP dissociates, your enzyme is still inactive. Now, when this thing is not occupied, when the site is not occupied by GDP or GTP, that means it can be occupied again by a GTP molecule, okay? Because it has more affinity for GTP. Then your enzyme is going to be active again. Okay, so it does on and off type of switch. Okay, so let's see here. All right, so you have this enzyme that is a GTPase, right? That can break your GTP. Sometimes it does it on its own. Sometimes it needs a little push from this activating enzyme. Okay, so you have enzymes that are going to push your original enzyme to get deactivated. Okay, so if you put a little picture here, so this is your protein bound to GTP right there. So let's put GTP right here. So the enzyme itself has a GTPase activity, but the enzyme sometimes cannot activate the GTP by itself. So it uses this GTP activating protein in order for it to cut a phosphate out of that GTP. Okay? So GAP is going to speed up the breakdown of GTP. Okay? When GAP works, then this whole thing is going to be connected to GDP, and this whole thing is going to be inactive. Now, when you have your enzyme that is empty, here, this is your pocket is empty, okay? So GDP came off, you can get GTP back in there and get the enzyme active again. Now, sometimes GTP goes in on its own. Some other times you need a little help from a helper enzyme that brings your GTP into the pocket. So this enzyme here that is helping, you called it guanine nucleotide exchange factor. Okay? So your GAF is going to bring your GTP into your pocket and is going to activate your G-coupled protein. Okay, is that simple? Should be? Yes? Okay, GAF is an exchange factor. It is going to go here and put GTP in there, GTP. Why exchange? Well, sometimes you have GDP sitting in there. Okay, if you have GDP sitting in there, GAF is going to go there, kick out GDP, put in GTP. Exchange factor. With GTP, that thing is going to be activated. All right. So, okay, I'm going to stop here because everybody's running away. All right, so I have one picture here. Pay attention to this guy here. You see this guy over there? Mind the gap. You have gap, you get inactivated. Got it? Maybe? All right, we pick up on Friday.